Hello, everyone. Welcome to today's webinar. I'm Kelly McSweeney, contributing editor at C&En Media, and I'll be moderating today's event. This webinar is titled Unlocking ADC Breakthroughs Through Payload Linker Chemistry Innovations and is sponsored by WuXi XDC. C&En works with sponsors to identify topics of interest and value to C&En's audience and consistent with C&En's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the top right corner of the screen or type your query into the Q&A box. If you are disconnected during the webcast, please log in again according to the instructions you received earlier. You're encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will pose as many as time permits. Please note that C&En does not endorse any company, products, or services that may be mentioned in the webinars, and that each webinar will be archived at C&En Online after the live webcast. Today's presentation is sponsored by WuXi XDC. WuXi XDC is a leading global CRDMO focused on antibody-drug conjugates, or ADCs, and the broader biconjugate market. It provides end-to-end contract research, development, and manufacturing services for biconjugates, including ADCs. Its services cover antibody intermediates and other biologics intermediates, chemical payloads and linkers, as well as biconjugate drug substances and drug products. During the presentation, we will hear from Jack Gangcheng Yang. Jack is a seasoned ADC and payload linker expert with extensive expertise in medicinal chemistry, protein chemistry, and the development of complex biotherapeutics, including ADCs. Over his 20-year career, he has contributed to groundbreaking therapies at leading organizations, including Wyeth Pfizer, MedImmune AstraZeneca, NIH, and CDMOs. Jack's work spans small molecule drug discovery, biologics characterization, and ADC development. At Wyeth Pfizer from 2002 to 2010, he established SAR models for lead optimization across diverse drug programs. Later, at MedImmune AstraZeneca and NIH from 2010 through 2019, he specialized in the analytical characterization of monoclonal antibodies, ADCs, and conjugate vaccines, playing a key role in the approvals of Imfinzi and Lumoxi. Since 2019, he has focused on ADC payload linker synthesis in the CDMO space, leveraging his deep knowledge of synthetic and medicinal chemistry to advance next-generation ADCs. And I will now hand it over to Jack.
Thank you, Kelly, for your warm introduction. I'm excited to be at C&En Medium today and to share what we have learned from CDMO service projects on how to enable ADC breakthroughs by payload linker chemistry innovation, including both the structure design and manufacturing process development. My presentation today includes 4 parts: strategies for payload linker research and development, a review of the conventional and new classes of payload linkers, our process strategies for large-scale synthesis of payload linkers, and at last, a brief overview of the integrated ADC services at WuXi XDC. First, let's see our support for payload linker research. This is for the discovery of preclinical ADC drug candidates, or PCC. In the context of medicinal chemistry for payload linker, the structure-property relationship is fundamental for finding the optimal ADC candidate. The selection of payload release mechanism is related to the design of MOA. Linkers may be either cleavable or non-cleavable, depending on the desired pharmacology profile. The physical-chemical properties of the payload may vary from one kind to another. They can be hydrophobic, such as MMAE, DM1, and exatecan, or hydrophilic, such as certain pegylated compounds. The method to attach to the antibody may include stochastic amide bond formation between lysine residues and active esters, thioether bond formation by microaddition of sulfur groups to a microacceptor such as a melamide linker, glycan-based conjugation, enzymatic conjugation catalyzed by enzymes such as sortase or MTG. Furthermore, the payload linker design must support precise control of DAR value and DAR distribution. Same as a small molecule drug discovery, the discovery of successful ADC candidates also requires iterative cycles of design, testing, and optimization. From a medchem perspective, when we work on a linker optimization, we should consider at least the following key factors. First, the physicochemical properties are important. Some polar or charged groups can help with the necessary hydrophilicity. The length and rigidity of the linker influence the PK of the ADC. The molecular weight and polar surface area of the linker should be balanced for tissue uptake. Second, the serum stability of the linker is key to ADC safety. For example, a steric hindrance adjacent to the cleaving site, multiple release mechanism, and blocking the proteinase using chemical moieties such as PEG are all medicinal chemistry strategies for the fine-tuning of the linker's serum stability. Third, adjustment of the kinetics of drug release is always necessary. The mechanism may involve tumor-specific enzymatic cleavage, extracellular triggering cleavage, or even non-cleavable designs. The kinetics of payload release should be tailored to the therapeutic context of the ADC molecule. Fourth, other chemical properties can also be important variables. For example, modifying the hydrophobic groups, adjusting the steric hindrance around the hydrogen bond donors, and using some branched structure for dual payloads can further refine the PK performance of the ADC. At last, the most appropriate linker structure must be determined case by case, according to the therapeutic context of the ADC molecule as a whole. For ADC, the payload is the therapeutic core. Its selection is driven by the intended MOA and may include microtubule inhibitors, nucleic acid modulators, protein degraders, radioisotopes, or immunostimulating agents, etc. The SAR study on the payload should focus on the identification of essential pharmacophores, structural modification, conjugation sites, and the feasibility of using dual payload to enhance efficacy, improve toxicity, and broaden the therapeutic window. In order to help our clients speed up their ADC programs cost effectively, we have developed processes for the synthesis of both conventional and new classes of payload linkers, along with the key intermediates for widely used payload linker combinations. Many of these compounds are available off the shelf from milligram to grams. Currently, we supply a comprehensive list of ADC payloads off the shelves. These include, but should not be limited to, the following. First, a variety of conventional microtubule inhibitors, such as auristatin derivative MMAE, MMAF, tubulysin series, methycin series like DM1, DM4, and complicated compounds like eribulin, etc. Second, most popular DNA modulating agents such as SN38, exatecan, doxorubicin, and PNU, etc. Third, some prevalent and specialized payloads such as BCL inhibitor, STING agonists, oligonucleotides, precision metal chelators, and steroid-based immunomodulating agents. In addition to the development of the synthetic methods, we have also registered drug master files for some repeatedly used chemicals at FDA. These drug master files are available for reference to support regulatory filing at FDA. The payload linker-related drug master files have helped our clients to save time in the course of R&D filing. For payload linker, the manufacturing needs some special capabilities. First, the facilities must be equipped to handle high-potent chemicals at exposure limit as low as 10 nanograms per cubic meter. Second, there must be a comprehensive CMC strategy and a technical expertise. Third, a rigorous API-grade quality system should be in place, and the quality system must be aligned with FDA or other regulatory expectations. For the design of ADC, the connector of the payload linker plays a critical role. It defines how the linker attaches to the antibody. It also determines what conjugation method to use. Commonly used connectors include, but should not be limited to the following categories. Number one, melamide is a connector that reacts with sulfhydryl on the antibody. It has been the most used one, such as the connector on Enhertu and a few other approved ADC drugs. Number two, active esters such as NHS esters are connectors that react with lysine amines. It was also in the approved ADCs, such as the one in Kadcyla and Besponsa. Number three, EBCO, BCN, and azide pairs are used in azide alkyne cycloaddition click reaction. This can be used for site-specific conjugation, such as glycan-based conjugation. Number four, GCO and tetrazine are also click reaction pairs by Deuce auto reaction. Some emerging ADCs are using it as an orthogonal click connector. It is also good for site-specific conjugation. Number five, hydrazine and aldehydes form hydrazone bonds that are pH sensitive. Number six, pyridine dione and vinyl sulfones can form thioether bonds with cysteine. Number seven, sulfonyl fluoride is also a connector. It reacts with the tyrosine residues and make a sulfonate ester linkage. Number eight, isocyanates and isothiocyanate can form either urea or thiourea bonds with lysine. At last, there are other strategies such as disulfide rebridging, phosphine azide, starting ligation, and so on. At WuXi XDC, we offer a wide range of such conjugation connectors, conduct a stability study, and GMP manufacturing of payload linkers with different connectors. Let's see the cleavable linkers commonly used in ADC. First, acid cleavable linkers such as hydrazone or cis-aconityl groups. These are designed to break down in acidic environment of the tumor cells. Second, enzyme cleavable linkers, including peptide-based designs like VC, GGFG, and FK. These are cleaved by lysosome proteins such as cathepsin B. Third, glutathione-sensitive linkers, typically disulfide-based, are cleaved in the reducing environment of the cytosol. Linker components can include peptides, PEGs, sugars, and branched structures for dual payload design. At WuXi XDC, our linker supply capabilities include a full range of cleavable and non-cleavable linkers, customer synthesis of new linker designs, and GMP manufacturing of payload linker products. To speed up ADC development, we have developed a method to make both conventional and emerging new payload linkers. Our inventory includes ready-to-use compounds such as VCMMAE SMCC DM1 and pegylated payload linkers. The next generation XDC payload linkers, such as chelators with linkers for antibody radionuclide conjugates, PROTACs with linkers, and oligonucleotide with linkers, and other novel formats. To support this innovation, we have built new payload linker enabling technology platforms, such as solid phase synthesis of peptides and oligonucleotides, chemical solid property and morphology study, flow hydrogenation and flow chemistry for critical reaction conditions, chemical catalyst screening, and biocatalysis for the synthesis of chiral payloads and linkers. These platform technologies allow us to deliver high-quality products at high speed. At WuXi XDC, we provide cutting-edge conjugation and payload linker technologies, along with strong process development expertise. First, we enable client technologies through CMC development. By the end of 2024, we had advanced over 10 client conjugation technologies to CMC manufacturing. Second, we have developed in-house proprietary technologies. WuXi DAR4 and DAR-X conjugation methods enable flexible DAR control with improved homogeneity. As of June 2025, 7 ADCs using DAR4 have entered clinical trials, including 1 ADC in pivotal phase. The quantity of one of the GMP batches reached 2 kilograms of ADC. Our proprietary X-link connector solved the maleimide stability issue. We have also developed novel payload linkers such as TOP1 inhibitors on hydrophilic linker structures. Third, we actively partner with external inventors and learn from the best, such as Aptiv, HeathLink, and Intocell, all formed a partnership with us. Such partnerships expand the range of ADC technologies that can become available to our clients. Some academic scientists have found a novel payload linker with interesting performance. For example, Professor Wei Liu of East China Normal University published a self-immolative linker that can attach to 2 payloads. This could be useful for ADC design. In another paper, he reported homocamptocecin and a modified exatecan. Both showed strong potential for cancer treatment. To help our clients access such new technologies, we collaborate with Professor Liu and will make some of the novel chemicals readily available. Clients can take advantage of such invention by working with us, regardless of the IP at research and development stage. Such collaboration speeds up the adoption of new technologies and will drive the breakthrough of ADC innovation. When we talk about ADC innovation, let's pay attention to the emerging dual payload ADCs. Dual payload ADCs can be constructed by either dual sites or single sites. Dual sites dual payload ADCs are made with 2 payload linkers. The 2 payload linkers have independent reaction handles for conjugation. WuXi DAR-X technology is compatible with dual site conjugation method. Single site dual payload ADCs use branched linker magnifiers that share a common reaction handle, such as the schematic structures at the bottom of this slide. Why dual payload ADCs? According to this recent paper, the additive or synergistic effect of 2 payloads with adjusted DAR values can improve the therapeutic efficacy, expand the range of the indications, and overcome the drug resistance. More specifically, synergistic killing effect is possible because dual payload ADCs may combine 2 drugs with different mechanisms, and they target different survival pathways of the tumor cells. Second, dual payload ADCs may broaden the therapeutic windows because it may improve the drug activity and balance the toxicity and efficacy by optimizing the dose ratio of 2 drugs. Third, dual payload ADCs may overcome drug resistance because even if the tumor becomes resistant to one payload, the other payload could still have killing effect. At last, dual payload ADCs can regulate the tumor microenvironment using two drugs with different mechanisms. At AACR 2025, about 100 presentations were about new ADCs. Some have dual payloads and showed exciting preclinical or early clinical data. This table highlights the top 20 dual payload ADCs in oncology. 2 are currently in phase 1. Most others are under preclinical development. It is interesting that 2 Nectin-4 ADCs use 3 distinct payloads. This tells that there is a trend toward multifunctional ADC designs. Let's hope that some of these new ADCs will solve some unmet medical need in the near future. Recent advances in ADC design have used a variety of dual payload linker structures. Each of them has unique conjugation strategy and payload combinations. Structure-wise, currently dual payload ADCs have tried the following linkers. First, symmetric branched linkers for the attachment of 2 payloads. Second, asymmetric branched linkers for selective attachment to different payloads. Third, tandem linkers use 2 cleavable motifs for sequential or independent release. Fourth, dual click linkers use orthogonal handles such as DBCO and TCO for site-specific dual conjugation by click reaction. Fifth, hybrid cleavable and non-cleavable linkers such as GGFG and bromoacetol species or VC-PAB and SMCC species. In this paper, scientists worked on a trastuzumab-based ADC. They used the MTG-catalyzed conjugation between lysine amine and asparagine 297 to make a dual payload ADC. The DAR ratio between the 2 payloads was 2:1. Efficient, cost-effective synthesis of a high-quality payload linker is essential for ADC development, especially in GMP environment. To do this, the optimal process chemistry strategies are very important. Next, I would like to share some of WuXi XDC's experiences in this area, with a focus on CMC and GMP manufacturing of payload linker. As highlighted in this OPRD paper, ADCs are hybrid molecules that combine biologics and small molecule components. This dual nature brings unique challenges for the design and implementation of any comprehensive CMC plan. FDA also classifies ADC as a kind of combination product. Its regulatory submission follows the biologics guidelines. This requires integrated CMC planning across both biologics and small molecule modalities. For ADC, the small molecule part is the payload linker. As you know, the payload linker is often a long, flexible structure with relatively large molecular weight. This makes purification by crystallization challenging. Let's see some of the challenges of impurity specs for payload linker. Although the ICH guidelines for mutagenic impurities are not directly applicable to ADC drugs, it is essential to set proper impurity limits for payload linkers to ensure ADC safety. Without any ADC-specific regulatory guidance, phase-appropriate adoption of existing ICH standards can be a practical control. For example, the impurity control for payload linker intermediates may be less stringent due to the dilution effect during conjugation. Additionally, non-conjugating impurities are typically removed in downstream process, such as UFDF or liquid chromatography. Therefore, along the pipeline, based on the availability of tox data and the therapeutic index of the ADC, the impurity limits might be broader during the discovery stage and phase 1. With further study results down the pipeline, the specs can be more stringent. For example, in phase 3 and PPQ, the purity of the payload linker should be higher than 95 to 98%, and the structures of any process-related and product-related impurities should all have been confirmed. Because of the complex nature of payload linker synthesis, using a quality-by-design strategy is helpful for ROS development and important for the best selection of starting materials. To do this, the key considerations include, but should not be limited to, the following. First, optimize the process efficiency. Try to install the drug moiety in the later stages and minimize the number of high-potency steps. Second, install reactive groups in the later steps for impurity control. This will simplify your purification. Third, the physical properties should always be in your mind for easy isolation and purification. Try to avoid the use of any gas as reagent and the generation of oil-like intermediates. Fourth, cost and safety should be considered. Use expensive reagents in later steps. Avoid risky reactions such as high pressure, highly exothermic reactions. Fifth, choose convergent synthesis over linear ones. Six, consider the scalability of the reaction. Consider that the reaction is run at lower concentration as the molecular weight increases step after step. 7. Production footprint must be planned. Pay attention to the VTO and the PMI. The design of optimal ROS is necessary, but it is not sufficient. A proper QC strategy is very important. For a specific payload linker, the QC panel could be different. Besides the identity and the purity of the payload linker itself, the QC tester should also pay attention to some impurity-related items, including but not limited to: first, chiral impurities are often from starting materials or from racemization during the process. Second, analogous impurities, often from the polymeric components like polyethylene glycol or polysaccharine. A QC method should be ready for each of these kind of polymeric impurities. Third, reactive or unstable moieties, such as microacceptors, diffuse odor reactants, or some materials that are easy to be hydrolyzed. This table lists some specs for payload linker products, including the test items and the test methods. The specs for non-GMP and GMP productions are different. Overall, the general specs include appearance, identity, purity, impurity and residues, endotoxin and bioburden, and very often, the current purity should be reported. Endotoxin control is critical for payload linker of ADC. Compared to small molecule APIs, controlling endotoxin during payload linker production is more challenging. HPLC purification followed by lyophilization can enrich endotoxin rather than removing or flushing it. At WuXi XDC, our control strategy includes the following 7. 1. Minimizing endotoxin source through the GMP-certified raw materials. 2. In-process bioburden monitoring. 3. Purification techniques such as UFDF, chromatography, activated carbon, and endotoxin-specific adsorbents. 4. Analytical testing for product release and method validation. 5. Strict personnel and workflow controls. 6. Regulatory compliance is vigorously maintained everywhere. 7. Audit readiness, with all facilities aligned to ICH guidelines all the time. WuXi XDC has extensive experience in delivering payload linker CMC projects. The early phase payload linker project usually takes only a few months. As an example, a 15-step synthesis of a high-potent payload linker was completed within 9 months. Over 100 grams of GMP material with over 98% purity, accompanied by full QA/QC support, was transferred for conjugation. This demonstrates our capability to rapidly advance payload linker CMC project from process development to GMP manufacturing. For late-phase projects, we promise a 15 to 16 months CMC timeline. These include about 3 months for process block, 5 to 6 months for PPQ readiness, and 6 to 7 months for the production of 3 PPQ lots. This accelerated timeline is achieved through parallel execution of process research and development, analytical development, QC readiness, and regulatory support. Such internal collaboration ensures fast advancement towards clinical and commercial manufacturing. You may have noticed that the most frequently used word today is deliver. Yes, timely deliver to clients is key to our service because turnaround time is crucial for our clients. At WuXi XDC, mission will be accomplished no matter what. Failure is never an option. In the past couple of years, our success rate has always been over 98%. The advancement of technology has helped us deliver high-quality services. Here, I would like to share a few examples and tell you how technology breakthroughs have added value to some ADC programs. The first example is about the use of flow reaction. Comparing to batch reaction, flow reaction has advantages such as precise control, improved safety, higher yield, better reproducibility, and environmental benefits. We have proactively adopted flow technology in payload linker synthesis. The first case is linker daughter synthesis for ARC project. The benzyl protecting group need to be removed by hydrogenation. Traditional batch hydrogenation couldn't go to completion, always leaving about 30% starting material unreacted, and the yield was low. By switching to flow hydrogenation, we got complete conversion in a few minutes with good yield and high purity. The second case is the synthesis of a payload intermediate. We needed to reduce the azide group into amino group in a kiloscale reaction. However, the azide is a high-energy chemical. It could not be safe to run in large scale. We developed a flow hydrogenation and got excellent yield using minimum amount of palladium. The third case is to make a self-emulsive linker moiety. The nitrobenzene needs to be reduced to aniline with the F-mark intact. Conventional bench reduction always gives about 25% DF-mark side product, which is not needed. Flow hydrogenation solved this problem, and we got very good yield. The reaction ran at 8 kilograms a day to produce over 30 kilograms of product. Quite often, the freeze drying after HPLC is a bottleneck. After fractionation by HPLC, the pooled fractions add up to be a large volume. It is not easy to freeze dry such a big volume. Sometimes it takes over a month to get several hundred grams of payload linker by freeze dry. What's more, the modifiers of the mobile phases, such as formic acid or TFA, can both cause degradation of the payload linker. We have developed an on-column enrichment method. After pulling HPLC fractions, we load them onto a reverse phase column again. Large part of the water was let go. Hydrophilic modifiers were easily flushed with water. Then the payload linker can be eluted with a small amount of high organic solvent. The graph at the right side of this slide shows the volume change of 11 different batches of payload linker. You can see that the on-column enrichment can reduce the liquid volume by 80 to 90%. As a result, the enriched payload linker fractions can be lyophilized to dryness much faster. For example, to make 850 grams of a novel linker VC-MMAE, the time for freeze drying dropped from 40 days to just 5 days. A beautiful bonus with the enrichment method is that the removal of modifiers also improves the stability and purity of the payload linker. This method has been applied across multiple payload linker production campaigns. This technology has been presented at the World ADC Conference. If you are interested in knowing more details, please reach out. We will be happy to share the poster with you. We all know that a strong analytical support is very important for payload linker manufacturing. WuXi XDC has developed many analytical methods for in-process control, impurity profiling, and QC release. One example is the identification of conjugatable impurities. Some impurities, like these 4 structures listed at the top, carry reacting handles or connectors to react with the counterpart on the antibodies. In this case, they have either maleimide connectors or breakable disulfide. Both may react with exposed sulfur hydro groups on the antibody. Once the impurities are conjugated to antibodies, it would be hard to be isolated from the desired ADC, and it will affect the purity of the ADC drug substance, leading to potential safety risks such as toxicity interference. Therefore, it is necessary to recognize such impurities during the payload linker synthesis stage and try to prevent them from being carried on to the payload linker product. Briefly speaking, to recognize such impurities, our analytical scientists performed a mock conjugation using N-acetylcysteine as a model counterpart. Impurities that conjugate with N-acetylcysteine will shift conjuration time in the HPLC and makes it easy for being recognized. The distinction between conjugatable and non-conjugatable impurities help us to set more strict standards so that we can reduce the risk of interference from such impurities in toxicology studies. Finally, let's look at the strategic advantages of using the fully integrated CRO and CDMO platform for ADC development. WuXi XDC offers a fully integrated CRO and CDMO platform. Every part of an ADC molecule can be done on the same campus, including the payload linker synthesis, antibody generation, conjugation development, and drug substance, drug product manufacturing. This one campus design eliminates the delays and troublesome inter-CDMO transfer of materials and information. It also streamlines the communication between function groups and significantly shortens the time for every project. From the end of 2023, we have expanded rapidly based on the enable, follow, and win strategy. The increase of project numbers proves the advantage of this one-stop-shop service model. In 2024, discovery projects grew from 427 to 681 within 1 year, and 45 discovery projects have advanced to integrated CMC stage. There had been a significant increase across preclinical and clinical programs, and the accumulated number of integrated CMC projects reached 194 by the end of 2024. In 2024 alone, we signed 53 integrated ADC contracts, launched 23 new integrated CMC projects, and filed 30 INDs. Also, in 2024 alone, we have delivered more than 300 GMP batches of ADC. The number of PPQ and commercial projects also increased. The payload linker department is part of the integrated CDMO platform and has supported ADC projects across all phases. demonstrating strong expertise in this area. For preclinical development, the payload linker service includes molecule modification, synthetic route design, and process preparation. We have delivered more than 400 payload linkers, and 15 payload linkers are related to IND filing. For early phase development, the work scope includes route optimization, impurity synthesis, tech transfer, and GMP support. We have completed over 310 CMC projects, and over 30 of them went to international IND filing. It takes 4 to 6 months for one tox lot and one GMP lot production. So far, we have got 100% success rate. For late phase development, the work scope includes process lockdown, characterization, and potential genotoxic impurity profiling. It takes 6 to 8 months for process characterization, and currently 7 PPQ projects are going on. For ADC discovery services, we support from concept to preclinical candidate. Our integrated services include target evaluation, antibody discovery and generation, payload linker development, bioconjugation development, in vitro and in vivo studies, etc. We bring all these functions together in one company to speed up the ADC development. More specifically, for target discovery, we perform tumor-associated antigen evaluation and the selection from multiple angle analysis. For antibody discovery and antibody generation, we perform antibody lead identification, lead optimization, and production using WuXi XDC's state-of-the-art technologies. For payload linker synthesis, we have talked a lot today. We support ADC projects using our payload linker library for early stage screening, using medicinal chemistry and SER support for structural optimization, and based on strong process chemistry capabilities for large-scale synthesis, including manufacturing in a GMP environment. For bioconjugation, our teams have accumulated experiences for a variety of many conjugation methods, including but not limited to lysine-based conjugation, cysteine-based conjugation, glycan-based conjugation, enzyme-based, and non-natural amino acid-based conjugation. We also have our own proprietary conjugation and payload linker technologies, such as WuXi DAR4 and WuXi DARX, WuXi X-Link, etc. And we partner with external platform companies or research institutions to leverage novel technologies and expand our toolbox. We support in vitro biological studies, perform ADC-related assays such as ADC binding, ADC internalization, in vitro cancer cell killing and efficacy study, bystander killing effect, plasma and serum stability study, and metabolite identification, so on and so forth. We can support in vitro pharmacological studies too. This includes animal tests based on specific efficacy models, PK/PD, and exploratory toxicity assessment. Before the final selection of the PCC, we can perform the characterization of physical chemical properties and the developability potentials as well. In short, as long as a client comes with the idea, we help propose the exploratory roadmap and escort the project from concept to preclinical candidate, the PCC. With decades of CDMO service experiences and over 98% success rate for the delivery of clients' projects, WuXi XDC has become a top service provider for global pharmaceutical and biotech companies. By the end of 2024, we have over 500 global partners, more than 900 ADC or XDC projects. We have delivered more than 14,000 bioconjugate molecules by the end of last year and have helped global clients submit 85 INDs. We are proud to be the only CDMO with facilities for antibody production, payload linker synthesis, conjugation development, drug substance, and drug product manufacturing all together in one campus. Currently, we have 2 global manufacturing sites. Our second site in Singapore is scheduled to begin operation and services by the end of this year. This is the end of my presentation. Please keep our contact information on this page and reach out for collaboration soon. Please also contact us if you are interested in knowing more technical details of the case studies. Thanks for your attention. I can answer some questions now.
Thank you, Jack, for that excellent presentation. We do have some questions from the attendees. The first question is, does WuXi XDC have the capability to synthesize molecular glues, PROTAC, and other related protein degraders?
Yes, we have made PROTAC and PROTAC ADCs for our clients. PROTAC synthesis has similar technical requirements as payload linkers. It requires HPLC purification and freeze drying. Molecular glues are also categorized as protein degraders with molecular weight less than 500 Dalton. Same as the small molecule drugs, we can also make molecule glues for clients.
Thank you. The next question is, does WuXi XDC have any tools to rationally and quickly select dual payloads within WuXi XDC's payloads inventory and beyond?
Yes, we have a large inventory of payloads. Most of the known ADC payloads are stored in this inventory. Our chemists can make linker payloads based on client selection of dual-site conjugation or single-site conjugation with branched linkers or linear linkers.
Next question is, what is WuXi XDC's capability in peptides?
The payload linker department of WuXi XDC has an established platform for peptide synthesis with the capability of custom synthesis of peptides from milligrams to multi-kilograms with linear or cyclic structures. Currently, we have 300-liter solid-phase reactors. The batch size can go up to 1,000 grams with purity of over 99% pure by HPLC. We also have an analytical panel for peptide synthesis. The turnaround time for peptide with the amino acid length of about 10 to 60 can take about 1 month for most of the linker products. Most of the peptides that we synthesize are used for ADC payload linkers.
Next question is, what are the key considerations when selecting cleavable versus non-cleavable linkers for different payload mechanisms of action, or MOA?
Good question. There are several factors to consider, and there could be more. The MOA of payload and bystander effect are one of the concerns. DNA-acting payloads often need cleavable linkers. Tubulin payloads often need non-cleavable linkers. The PK and stability is also a concern. Non-cleavable linkers give higher plasma stability. Cleavable linkers risk premature release of the drug. Safety and efficacy is another concern. Cleavable linkers expand the efficacy but raise the toxicity risks. Non-cleavable linkers give a cleaner safety profile.
Next question is, what are the main challenges and solutions in scaling up payload linker synthesis from preclinical to commercial stages, particularly with complex multi-step processes?
I think this should be answered case by case, but there are some general challenges as well, including the handling of high-potent chemicals on a large scale, including the impurity control along the multi-step synthesis. The challenge to purify and dry a large quantity of semi-stable chemicals within a short period of time without significant degradation. Another challenge is the reproducibility of process in large scale.
That's all the time that we have. Any questions we didn't get to answer during the webinar will be answered over email after today's live event. Thank you again, Jack Gungcheng Yang, for your fascinating presentation. And thank you, participants, for being a great audience. Be sure to check C&En or C&En Online for information on the next edition of C&En webinars. Thank you, On24, for technology and production services. And thank you, Wuxi XDC, for the sponsorship that made this interactive webcast possible. For C&En webinars, I'm Kelly McSweeney. Goodbye.